Hello everyone! Today we are going to explore a very fundamental concept in histology and cytology, namely the fixation of tissues. Before we begin, let me ask you a simple question. Have you ever wondered how delicate biological tissues can be preserved in such a lifelike state for microscopic examination? The answer lies in the process of fixation, which forms the foundation of all histological techniques. In this video, we'll explain what fixation is, why it matters, the main aims, common fixatives, and practical examples you can relate to. Follow slow-paced narration with clear animated diagrams and captions to make each step easy to follow. Let's begin with a simple definition and build from there to practical guidance you can use in the lab. Fixation is a process by which cells or tissues are preserved in a chemical and partly physical state so that they can withstand further processing with various reagents without undergoing decomposition or distortion. In simple terms, fixation locks the tissue in a condition that closely resembles its living state. This is extremely important because once tissues are removed from the body, they are prone to rapid degradation due to enzymes and bacterial activity. Proper fixation preserves morphology and cellular detail, so a pathologist can accurately interpret structure and disease-related changes. Fixation also preserves chemical and molecular targets so special stains and immunological tests can detect specific proteins or nucleic acids. Without reliable fixation, microscopic interpretation becomes unreliable and diagnostic accuracy suffers. Now, let us discuss the aims of fixation. Aim 1, to preserve tissue structure and prevent autolysis, keeping cells and organelles intact for accurate morphology. Aim 2, to stabilize proteins and cellular components so they resist damage during processing and staining steps. Aim 3, to maintain relationships between cells and extracellular structures, preserving tissue architecture and spatial context. Aim 4, to render tissue compatible with later reagents, embedding media and sectioning so processing can proceed without artifact. Aim 5, to retain molecules of interest for special stains and immunohistochemistry whenever possible, while minimizing loss of antigenicity. Fixation begins immediately after tissue removal to minimize post-mortem changes. Rapid fixation preserves delicate features and prevents autolysis. Primary practical step is immersion of appropriately sized tissue pieces into an adequate volume of fixative to ensure penetration throughout. Many chemical fixatives work by cross-linking proteins, forming stable bonds that lock structures in place and resist later processing. Other fixatives, such as alcohols, act by coagulating or precipitating proteins through dehydration rather than forming cross-links. Common routine fixative is 10% neutral buffered formalin for light microscopy and glutaraldehyd for electron microscopy to preserve ultrastructure. Choice and duration of fixation depend on downstream tests, sample size, and the balance between morphology preservation and antigen retention. Practical example: a small biopsy placed in formalin at a ratio of at least 10 parts fixative to 1 part tissue helps ensure consistent fixation. Avoid very large samples or thick pieces that impede fixative penetration and create underfixed central regions with artifacts. For immunohistochemistry, fixation may require milder conditions or antigen retrieval later to preserve target molecules for antibody binding. Common pitfalls include delayed fixation, inappropriate fixative selection, insufficient volume, and inadequate fixation duration. In summary, fixation locks biological structure, prevents decomposition, and prepares tissue for accurate microscopic study and reliable testing. Thank you for watching. Apply these principles to improve sample quality and diagnostic reliability in your histology practice. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.